Let us learn instrumentation techniques of NMR spectroscopy. NMR spectroscopy instrumentation involves a strong magnetic field, radio frequency pulses, and a detector to analyze atomic nuclei and obtain structural information of any molecule. Key components include a magnet, radio frequency transmitter, a receiver, and a computer for data processing. NMR spectroscopy works by varying the machine's emitted frequency over a small range while the sample is inside a constant magnetic field. A detector then interprets the results and sends it to the main console, a computer that analyzes and records the data. The NMR spectrum provides information about the chemical environment of the nuclei, allowing for identification of different types of atoms and functional groups in a molecule. Nuclear magnetic resonance spectroscopy is a powerful analytical technique used to characterize organic molecules by identifying carbon-hydrogen frameworks within molecules. Two common types of NMR spectroscopy are used to characterize any organic structures. Proton NMR is used to determine the type and the number of hydrogen atoms in a molecule. Carbon-13 NMR is used to determine the type of carbon atoms in the molecule. The source of energy in NMR is radio waves, which have long wavelengths and thus low energy and frequency. The NMR spectrophotometer consists of the following components: sample holder, permanent magnet, magnetic coil, radio frequency generator, radio frequency receiver, readout system, that is a computer, and the detector. This is the pictorial diagram of NMR spectroscopy. So we have a sample tube in which the sample is there and which is sandwiched between magnet. It can be an electromagnet or supermagnet or permanent magnet. And when magnetic field is on, the nucleus will precess and this will produce some signals, some frequency. And this frequency, if it is matches with the radio frequency transmitter, we will have a resonance. And this resonance frequency will be detected by a detector. And at the end, it will be fed into the computer and we can have an NMR spectrum. This is the diagram in the lab. So we have a sample holder and sandwiched between two magnets, and we have a radio frequency generator. And when there is a resonance, and the resonance frequency will be observed by a detector, and it will be fed into the system, LCD or a display system. Sample holder. The sample is placed inside the probe, which is located at the center of the magnet, in which the magnetic field is strongest and most homogeneous. The probe contains coils that both transmit the RF pulses and detect the signals emitted by the excited nuclei. The sample holder of NMR is normally tube-shaped and is therefore called the sample tube. The tube must be intransparent to RF radiation, durable, and chemically inert. Glass or Pyrex tubes are commonly used. These are steady, practical, and cheap. They are usually about 6 to 8 cm long and 0.3 to 0.5 cm in diameter, with a plastic cap to contain the sample. This type of nuclei is used for containing spectra of bulk samples and solutions. Permanent magnet. The core of an NMR spectrometer is a strong, stable, and homogeneous magnetic field. Permanent magnet or electromagnet or superconducting magnet all can be used in an NMR instrument. It should give stable and homogeneous magnetic field at 60 to 100 MHz. the strength and direction of magnetic field should not change point to point. Modern NMR instruments use high field magnets over 20 Tesla to achieve high resolution because the chemical chips are proportional to the magnetic field strength. The magnet size is 15 inches in diameter. This is a relationship between the resonance frequency. Third one is magnetic coil. There is a relationship between the resonance frequency of nucleus and the strength of the magnetic field in which the sample is placed. The relationship goes like this. So V equals constant multiplied by the external magnetic field H0. For the nucleus to resonate, the precessional frequency of the nucleus must be equal to the applied RF radiation. If the H0 is constant, the precessional frequency is fixed. Fourth one is radio frequency generator. It generates radio frequency pulses that excite the atomic nuclei into a higher energy state. In order to generate radio frequency radiation, radio frequency oscillator is used. To achieve the maximum interaction of RF radiation with the sample, the coil of oscillator would be around the sample container. The oscillator irradiates the sample with the RF radiation. The frequency of the RF pulse is tuned to the specific nuclei being studied. Example, proton or carbon-13. The oscillator coil is perpendicular to the applied magnetic field. A radio transmitter coil that produces a short, powerful pulse of radio waves 4 to 900 MHz. Fifth one is radio frequency receiver. It detects the weak signals emitted by the nuclei as they relax back to their original energy state. It is installed perpendicular to both magnetic field and the oscillator coil. It is tuned to the same frequency as transmitter. When the precession frequency is matched with the RF radiation, the nucleus induces EMF in detector coil, and this signal is amplified and sent to readout system. The receiver must be highly sensitive to detect these weak signals. Seventh one is detector. It detects the radio frequencies emitted as nucleus changes to a lower energy level. A detector then interprets the results and sends it to the main console. And sixth one is the readout system. We have changed into the topics. You can change into 7 and 6, 6 and 7. The readout system gives a spectrum as a plot of strength of magnetic field on x-axis and the strength resonance signal on y-axis. The strength of the resonance signal is directly proportional to the number of nuclei resonating at a particular field strength. A computer that analyzes and records the data. The NMR spectrum provides information about the chemical environment of the nuclei, allowing for the identification of different types of atoms and functional groups in a molecule. Now let us discuss NMR in chemical industries. So they are the applications of NMR in chemical industries. NMR spectroscopy has a wide range of applications in various fields, including chemistry, biochemistry, pharmaceuticals, and material science. First one is structure determination. NMR spectroscopy can provide detailed information about the molecular structure, including bond connections. Petroleum industry. One can characterize the crude oil and petroleum products and thereby determine the composition, molecular structure, and physical properties of hydrocarbons. Chemical analysis. It is used to determine the composition and purity of chemical compounds, including complex mixture, drug discovery and development. NMR plays a crucial in the identification and characterization of a drug molecule. One can study their interactions with the target molecules, aiding in drug discovery and optimization. Material characterization. NMR can be used to study properties, structure, and behavior of various materials such as polymer, ceramics, catalysts, and nanoparticles, contributing to advancement in material science and engineering. Metabolomics. It is used to identify and quantify metabolites in complex biological samples, and it is applied to understand the metabolic pathways and identify biomarkers for various diseases. Quality control in food and beverages. It is used to analyze the composition of food and beverages, including detecting adulterants, assessing nutritional content, and ensuring product quality and authenticity. Environmental studies. It is used to analyze pollutants and contaminants in air, water, and soil samples, aiding environmental monitoring and assessment. Environmental monitoring. It is used to monitor pollutants, trace metals, and contaminants in environmental sample, contributing to environmental assessment and remediation efforts. Forensic science. It is used to identify and analyze the unknown substances found at crime scenes and trace the evidences. Study of biological macromolecules. It is used to investigate the structure and dynamics of nucleic acids, DNA and RNA, other biological macromolecules, providing insights into their functions and interactions in living organisms. Polymer chemistry. It is used to characterize the structure, composition, the molecular weight of polymers, aiding in the development of quality control of various plastics and synthetic materials. Natural product analysis. It is used to identify the structural elucidation of natural products such as plant extracts and secondary metabolites, which often have pharmaceutical and biological significance. Isotope labeling studies. It is employed in studies involving isotope labeled compounds, which offer unique insights into molecular interactions and dynamics. Reaction monitoring. It is used to monitor chemical reactions in real time, providing information about reaction kinetics and intermediates formed during the process. Protein ligand interactions. It is used to study the binding interactions between the proteins and the ligands, aiding in drug design and understanding molecular recognition process. Quantitative analysis. It determines the concentration of specific compounds in a mixture without the need for extensive sample preparation. NMR crystallography. It combines NMR data with X-ray crystallography to obtain detailed structural information about crystalline materials. Geochemistry. It is used to study the geological materials such as rocks and minerals to understand their composition, structure, and environmental history. Imaging of porous media. It is used to image and characterize the porous materials like rocks and soils, helping in applications related to oil exploration, groundwater studies, and soil science.